Hello, Greeting International. Today we are gonna talk about DNA replication. This is in your book, chapter 8.3 from page 221 to 224. So, let's start. First, we need to remind you with the cell division. The cell division is a process in which a cell is dividing till it gives us two daughter cells identical to the original one. This cell division is undergoing among four main stages: G1, S stage, G2, and M stage. The most important stage that will support my session today is S stage, which means DNA synthesis. By the way, DNA replication is equal to DNA synthesis, the same meaning. In S stage, DNA is copied or replicated. As you see, original DNA is duplicated to give us two identical copies of DNA. Here in cell division, this is the original cell, this is the DNA inside. The DNA keep dividing till it gives us two copies of DNA, one copy in each new cell. We need to remind you too with the structure of DNA, which was in section 8.2. DNA is a double helix that are connected together with the nitrogen containing base. Here in more details, these are the two backbones of the DNA. This is the original strand, this is the complementary strand, and they are connected together with the nitrogen containing bases by a hydrogen bond, which is a weak bond that is easy to be broken down. The DNA replication is dependent on these facts. DNA replication is like unzipping a zipper. This is a zipper that consists of two strands or two sides. This is the DNA that consists of two sides. A zipper is connected from inside. The DNA too is connected from inside. So DNA replication is like unzipping the strands of the DNA to separate the two strands. Zipping and unzipping the DNA strands are making by enzymes and proteins. The most common enzymes are called DNA polymerases. DNA polymerases. DNA polymerases are enzymes that form bonds between nucleotides during replication. So enzymes unzip the double helix as you can see here. So this is the original DNA, and this is the enzyme that starts to break down the weak hydrogen bond that between the nitrogen containing bases. As you see that this enzyme are breaking down the DNA at specific places. These places we call it origins. These places we call it origins. Okay, so it's break down the two strands or the bonds between the two strands and give us this separated strands. Let's take a closer look. Please don't be panic. This is so easy, but just pay attention to me. Okay, this is the original DNA. This the brown thing is the enzymes that are working on breaking DNA and making new strands. This is the original strand. This is the complementary strands, and we are calling these two original strands a template. This word, a template. Template means something like a model to make or to help to make new strand completed. So this is the strand. It starts to make a new DNA in this direction, and we all know that this strand is a complementary or opposite to the original strand. So anything happen in this strand, the opposite is happening in this complementary strand. So the direction of making new DNA in this strand in this way, so the direction of making new DNA in this complementary strand will be in the opposite direction. We have two types of strands. Here we have two types of strands. We have this strand that's called leading strand, and we have this strand that's called lagging strand. So what is the difference between them? In this picture, we will see here is the leading strand. As you see, the DNA replication is occurring in a continuous way in one direction. It's one piece, okay? But in lagging strand, this is the lagging strand. What is the difference? Can you see the difference between them? Yes, this DNA replication in one piece, and here is a space. Here's a DNA replication, one piece, and here is a space, one piece, and here is a space. We call it a lagging strand. A lagging strand is a strand in which the DNA replication is occurring in a discontinuous piece by piece way, as you see here. So this strand is called, we called it a leading strand, but this strand we called it a lagging strand. So why all this? Why all these complicated processes? Why we are duplicating DNA? We are duplicating DNA in a simple way for growth and development. You need to grow, man, and repair. We have a a serious or a simple wound. You need to be, this wound needed to be repaired. And the most important thing, reproduction. DNA, as we all know that DNA carries the genetic information. This genetic information are passed from generation to another. You can do this until the DNA is replicated. Thank you for your time.